Hello, grade 12 students. In this video, we're going to talk about the nervous system. And to be more specific, we're going to talk about chapter 9 about the functions of the neurons. So, chapter 9 about the functions of the neurons. So, prepare yourself in order to start with first document about the resting potential. So, document 1, resting potential on page 1A2. Already we discovered all of these in the previous PowerPoints and the previous videos. So now we're going to move to our book in order to start some applications and some experiments. So here in document 1, resting potential. The potential difference that exists between the inside and the outside of the membrane of a non-stimulating neuron is called resting potential, or it's called a membrane potential. What is the origin of this potential and how it is maintained? So, presence of resting potential by experimental verification. To prove the presence of resting potential, recordings are obtained on a giant axon of a neuron by using microelectrodes connected to a sensitive galvanometer or an oscilloscope. Insert into a giant axon of a squid a glass microelectrode which contains a KCl solution with a very thin tip having a diameter less than 0.05 micrometer in order not to damage the membrane. This microelectrode is connected to a plate at the top of the oscilloscope, while at the bottom of the oscilloscope, the other electrode is connected to a plate immersed inside a physiological solution like seawater that surrounds the nerve fiber. So upon introducing the microelectrode in the interior of the nerve fiber, and here we have both of them are in the exterior, so we have 0 millivolt. The second, whenever we have one in the interior and the other in the exterior, we have a abrupt downward deflection of the electron beam appears on the screen of the oscilloscope. So it moves here. This is external electrodes. Here we have the potential difference was 0. Whenever we explain one in the internal and one in the external, so we have the drop in the potential difference to -70, reaching the resting potential. So here, after we connect the electrodes of the galvanometer, the needle deviates, which indicate that we have here the deviation of the needle, indicate that we have polarity. This polarity of the nerve fiber is at rest. So after connecting the electrode, inserting one in the intracellular medium, keeping the other in the extracellular medium, the potential difference decreases to -70, which indicates that we have a polarity and it's equal to -70 millivolt. Origin of a resting potential. The plasma membrane of a neuron, document C, separates two liquids compartments, the extracellular and the intracellular medium, whose ionic composition is represented in document D. Here we have document C, the structure of the plasma membrane. We have the phospholipid bilayer. This is the cholesterol, and this is the protein with the pore. Here we have the interior and the exterior, and we have the cell membrane. So in order to better understand the origin of the resting potential, the following experiment is performed in document E, where we have two compartments, 1 and 2. They have different ionic composition, are respectively similar to intra and extracellular medium of the mammal's neuron. These compartments are separated by a permeable membrane. It's permeable only, permeable only to K+ ions. Two electrodes placed on the two sides of the membrane record a potential difference of 80 millivolts. K+ ions, they diffuse. They move from medium 1 to medium 2 following the concentration gradient. They move from a medium high concentration to medium of low concentration. This flow of cations lead to an electrical change. Side 1 of the membrane becomes more electronegative since it loses positive ions with respect to side 2. This electric field increases as the flow of the K+ increases from 1 towards 2. At the end of an equilibrium is established between the force of the diffusion and the electric force. The potential difference that is measured represents the equilibrium potential of the K+. To stimulate the fact that the plasma membrane is permeable, the membrane M is replaced by membrane M', which is very, very permeable to K+ and slightly permeable to Na+. the Na plus and K plus diffuse in opposite direction according to their concentration gradients, but with a more rapid diffusion of K plus than that of Na plus ions, as shown in document F. So more K plus will diffuse than Na plus. This is shown in document F. This document. Okay, so as the concentration of Na plus is higher, how to maintain the resting potential? As the concentration of Na plus is higher in the extracellular than in the intracellular medium, and inversely for K plus, the ions diffuse in a small quantity through the membrane. If the diffusion continues, the internal and the external medium would have the same concentration. However, the state of electron neutrality is never reached in the living cells. And to understand the mechanism responsible for maintaining this membrane potential, the following experiment is performed. An isolated axon of a squid is placed in a seawater to which radioactive Na plus ions are added in order to measure the flow of Na plus. DNP, dinitrophenol, is added to the medium. This blocks the metabolism by inhibiting the phosphorylation of ADP. What is ADP? It's adenosine diphosphate. It blocks its phosphorylation into ATP. What the meaning of phosphorylation? Phosphorylation is addition of phosphate. ADP, it's adenosine diphosphate. ATP, adenosine tri. So di it's 2, tri it's a 3. So it inhibits the phosphorylation, the addition of phosphate to ADP. A gradual decrease in the outflow of Na plus is observed until it stops completely. ATP is then provided to the axon, which is still under the effect of DNP. Na plus are observed to flow out again across the membrane. The same results are obtained with the flow of K plus when radioactive potassium is used. In a living cell, the ionic equilibrium is never reached. We don't have an ionic equilibrium. This is because of the mechanism that pumps Na plus from the actively inside to the outside and allows penetration of K plus inside. Again, the concentration gradient. The system is called Na plus K plus ATPase, or it's called Na plus K plus pump. It's called sodium potassium pump. The principle is illustrated in document H. This is the principle of K plus, Na plus K plus pump. The protein molecule Y, this is the protein molecule Y, code transporter, carries Na plus from the internal to the external. Once outside, Na plus will detach from the Y protein. This molecule undergo conformational change. It changes from Y into X. So this X carries K plus from the external to the internal. At this stage, the transporter X is detached from K plus, and then it goes back to the Y structure or the Y form. That again attaches to an A plus, and the process continues. This conformation change from X to Y, it requires energy provided in the form of ATP as shown here. Here we're going to solve the probability activity. Number 1 and number 2, we're going to solve them together. Number 3, number 4, and number 5. So after connecting the electrodes of the galvanometer, as I said, to the nerve fiber, the needle deviates, which indicates a polarity existing between the membrane of the nerve fiber at rest. After connecting the electroding of the oscilloscope and inserting one of the electrode into the intracellular medium while keeping the other one in the extracellular medium, the potential difference decreases to minus 70 millivolt, which indicates that polarity exists between both sides of the nerve fiber. Compare document D, the concentration of Cl- in the intracellular medium of the nerve fiber of a marine animal, 550 millimole per liter, is less than by approximately 11 times than that in the extracellular medium, 560. The concentration of Na plus, and you can use it for concentration, in the extracellular medium of a nerve fiber of a marine animal was 50 millimole per liter, which is less than by 9 times than that in the extracellular medium. which is 440, and here we have 50. The concentration of Na plus in the intracellular medium of nerve fiber of a marine animal was 400 millimole per liter, which is greater than by 20 times than that in the extracellular medium, which is about 20 millimole per liter. So this indicates that there is unequal distribution of ions on both sides of the nerve fiber. So after diffusion of K plus from one compartment to another through membrane AM and maintaining an equilibrium, a potential difference exists between both compartments equal 80 millivolt. However, when membrane AM is replaced by membrane AM prime, which allows the diffusion of K plus with a higher rate than the diffusion of Na plus, a potential difference is recorded between both compartments after ionic equilibrium exists equal to 70 millivolt. What does this indicate? This indicates that the unequal distribution of ions between both sides of the membrane and the selective permeability of the nerve fiber, allowing the outflow of K plus more than the inflow of Na plus, causing the appearance of resting potential. Number 4, experiment document 3. Before adding DMP, which inhibit the phosphorylation of ADP into ATP to medium containing radioactive Na plus ions, an outflow of Na plus from the intracellular medium to the extracellular medium. So after adding DMP and inhibiting the phosphorylation of ADP into ATP, no energy. So the rate of outflow of Na plus decreases and then stop. This indicates that the outflow of Na plus need more energy in the form of ATP. The same explanation for K plus, this is for outflow for Na plus, inflow for K. Last one, importance of Na plus K plus pump. It permits the pumping of K plus and Na plus, again the concentration gradient, which keeps the concentration of Na plus in the extracellular medium more than the concentration of Na plus in the intracellular medium, and the concentration of K plus in the intracellular medium more than the concentration of K plus in the extracellular medium. This causes continued inflow of Na plus and outflow of K plus with the concentration gradient, which causes the appearance of the resting potential minus 70 millivolts. So as a remark, what is diffusion? Diffusion is the flow of substance, as I said, from a medium of high concentration to a medium of low concentration with the concentration gradient. Passive transport is the transport that takes place with the concentration gradient. It doesn't need energy. While active, it takes against the concentration gradient, it needs energy. ATP, source of energy used by the cell. So ADP, adenosine diphosphate, upon phosphorylation, when I add phosphate, it becomes ATP. So adenosine triphosphate. Also, by the process of dephosphorylation removing P, it becomes ADP. Okay, guys?